Okay, so our next non-Mendelian inheritance pattern is going to involve different types of dominance. So we're going to look at incomplete dominance, and then we're also going to look at codominance. And the example that we're going to use is cows because I love cows. I think they're really cute. So we're going to use cows because I like them. So let's look at this scenario where we have some true breeding parental cows. True breeding, remember, means that they are, in fact, completely homozygous for their genotype. And I have a cow with black fur and I have a cow with white fur. And then we're going to look at these possible offspring down here. Aren't those cute little cows? So let's first start with what we know, and we're going to start with this complete dominance inheritance pattern that we're familiar with. So let's say we had these two cows, the black-coated cow and the white-coated cow, and all of my offspring would show this black coloration. That would be complete dominance. And that occurs when the phenotype of the heterozygote and the homozygous dominant are completely identical. So in that scenario, we would have the black cow having the genotype of capital B, because that cow is true breeding, and then this cow would be homozygous recessive. And I know that they're homozygous for both because these are true breeding cows. I know that the black coat would be dominant because we see that all of my F1 offspring would have the black coat. So this cute little cow, its genotype will be heterozygous. So that's what we've known so far. We've known complete dominance. But we're going to come back and we're going to look at different scenarios. So let's say that we did this genetic experiment again and we did not get any black cows from that mating, but instead we got this fluffy little cute brown cow. So let's come back and erase these genotypes because this is not showing complete dominance, so we're going to denote alleles a little bit differently. A blending, appearance of blending between black and white is brown. Brown's kind of in the middle of the spectrum of those coat colors. So when the phenotypes appear to show that blending, we say that that's incomplete dominance. The phenotype of the F1 hybrid is somewhere between the phenotypes of the parental varieties. So that's going to look kind of different because one allele isn't completely dominant over the next. We show like kind of a blending here. So how we would denote that is quite different. I can't make one capital and one lowercase. So what I'm going to do is I'm going to have one letter for coat color for both of those cows. And then I'm going to use the superscripts just like I did for sex linkage. So whenever there's something weird, if it's not just simple complete dominance, Mendelian genetics, I'm going to use superscripts. So I'm going to use C superscript B for the allele for a black coat, and I'm going to use C superscript W for a white coat. One allele is not dominant over another. They're incompletely dominant, so I can't make one capital, one lowercase because there is not complete dominance shown because we have this phenotypic blending. So the genotype of this brown cow would be C superscript B, C superscript W. If you chose to write the genotype this way, that's completely valid as well because one allele is not dominant over the other. So either one of these genotypes would be appropriate for this brown cow. And this inheritance pattern would be incomplete dominance. So that is another option. But let's go back to this mating again between the black cow and the white cow. And let's say that we don't get any of the fluffy brown cows, but instead I get this cow that is spotted, white and black spotted cow. This is an example of codominance. Both alleles are dominant and we see each phenotype, but they are both expressed equally. So see here in this brown cow, there's like a blending of this phenotype. That cow isn't black, it isn't white, it's kind of an intermediate between those. But here we do see black fur and we see white fur. So this would be codominance. Both of those alleles are dominant. So we keep the same notation pattern. The C with the superscript B, the C with the superscript W, because they are both dominant, they are both expressed equally in this offspring. So this genotype for this spotted cow would be the same as the brown one. And you could write it either way because they are codominant, they're both expressed. But the main thing that you need to know is the difference between incomplete and codominance. We show a phenotypic intermediate in incomplete dominance, and in codominance, both of those phenotypes are expressed. Here's an example of incomplete dominance using plants. Here we have 4 o'clock flowers, and notice here that we have our parental phenotypes. We have a red flower plant and a white flower plant. All of the offspring here, the F1s, are showing pink. That is that intermediate phenotype, some type of blend between the phenotype of red and white. So we're going to denote the red allele as C superscript R, the white as C superscript W, and then our F1 offspring would all show this pink phenotype, so their genotype would be C superscript R, C superscript W. They would be heterozygous. So one thing that I want to talk about is when we're looking at incomplete dominance and codominance, sometimes it appears differently depending on how close we look at the example. If we're looking at the organism, if we're looking at a cell, if we're looking at the enzyme or the protein that's created from those genes. So let's talk about an example of sickle cell disease. Sickle cell disease affects many African Americans, 1 out of 400, and that is caused by a single substitution of one amino acid in the hemoglobin protein. So what I want you to notice is this is a notation that you are going to see very often. This HB stands for hemoglobin, and notice that they're using a superscript A, and that allele will produce normal blood cells, but the superscript B will produce sickled cells. So the heterozygote has sickle cell trait. So if we're looking at the organism, at the human being, someone who is homozygous for HbA will have all normal traits. Someone who has HbS and is homozygous for HbS will have sickle cell disease. But then if we look at the heterozygote, it seems to be an intermediate phenotype, because if you have sickle cell disease, you have many symptoms like pain, anemia, and that anemia is from having too few blood cells, which means that you don't transport oxygen appropriately, meaning that you're tired and dizzy and lightheaded, and affects growth and infection and many, many other symptoms that range from mild to very severe symptoms. That's if you're homozygous for HbS. So this would be that individual's genotype. But if we look at the heterozygote, that would mean that they have one HbA allele and then one HbS allele. Those individuals have sickle cell trait. Now, sickle cell trait could result in some pain episodes in various scenarios where they would be extreme, where low oxygen levels might be present, or they're dehydrated, or high altitude levels where the oxygen is not at a high concentration. But also, oftentimes athletes who have sickle cell trait are more likely to experience like heat stroke and muscle breakdown when they're competing at that high level. So now very often you'll have college level athletes who need to be tested for sickle cell trait because they are now more aware that athletes at that high level could experience some of these episodes that they're unfamiliar with because they didn't know that they were heterozygous for sickle cell. So at the organism level, that appears to show incomplete dominance, right? Where we have people who have two normal alleles, people have two sickle cell alleles, and then that people who have sickle cell trait who are heterozygous kind of have intermediate phenotypes. So it appears at the organism level like incomplete dominance. But if I'm looking at the cellular level, here I have individuals that are homozygous for the HBA alleles, and look at their red blood cells. Every single red blood cell is fat, and we have like this biconcave disc where there's this little like divot here in this red blood cell, and oxygen likes to sit right here attached to the hemoglobin protein. So the hemoglobin protein sits right here and it attracts oxygen, and oxygen sits right there in that biconcave disc. If I look at people with sickle cell disease, remember sickle cell disease, those individuals are homozygous as well. They do not have any of these normal red blood cells. Instead, their cells are sickle shaped. They look like the letter C. And we do not have that little kind of divot right there where oxygen likes to sit and be transported. So there's going to be issues with the transport of oxygen. And it also leads to things like pain because these sickle cells get stuck in capillaries and we don't have movement of blood flow very well. Now we're going to look at people with sickle cell trait. These are our heterozygotes that have both alleles. And notice here, this is an image of someone's blood who has sickle cell trait. They have normal red blood cells, but they also have sickle-shaped cells. So if I'm looking at their blood, that looks like complete dominance to me, right? We show normal and sickle cell. It's kind of like that spotted cow. We have black and white coat. So at the cellular or molecular level, this allele looks like it's codominant. But if I'm looking at the person, if I'm looking at the organism, it looks like incomplete dominance. So as you're going through and answering questions about is this showing complete or incomplete dominance, you just need to pay special attention to the prompt as you're reading it and see if they're showing you something that's incomplete or codominance at the organism level, or if they're talking about something that is smaller like the cellular or the protein level. Here's another example. Here we have Tay-Sachs disease. Tay-Sachs disease is mostly prevalent in Jewish populations, and this is a fatal disease because it affects enzymes that break down accumulations of lipids. So if those lipids accumulate in neurons, it can lead to many disorders of those neurons, and it leads to death without that functioning enzyme. So if we look at the organism level, someone who is homozygous dominant or someone who is heterozygous will have no effects at all. Like, they're going to have no accumulation of lipids. They're going to make enough enzyme to get rid of those lipids from the brain, and they are fine. So at the organism level, Tay-Sachs disease appears to be recessive. You're only going to show that lethal phenotype if you're homozygous recessive. However, if we look at the biochemical level, what we notice is that if you are homozygous dominant, your enzymes that remove those lipids are working at 100%. If you have two alleles that are different, you're a heterozygote, those enzymes are working at a 50% capacity because this dominant allele makes a functional enzyme, this recessive allele makes a non-functional enzyme. So only half of your enzymes are functioning, but 50% is good enough to get rid of those lipids that accumulate in the brain. but we're showing an incomplete dominance here at the enzyme level. And then finally we would have the homozygous recessive individual, and those enzymes don't work at all. So here, if I'm looking at Tay-Sachs disease, if I'm looking at the person, at the organism, that appears to be a dominant trait. You're either dominant or recessive at the organism level, but if I drill down and I look at the enzyme activity, if I look at the biochemical level, that looks like incomplete dominance to me. So what we now know is that dominance and recessiveness, it's a continuum, and it really depends on what we're looking at. Are we looking at the organism? Are we looking at the cell? And then we need to drill down and actually look at the protein that is created.